I'm Marina Sanz. And the first thing I have to say is that I'm doing my PhD in Spain, in Madrid, in the Universidad de Juan Carlos, in a pharmaceutical lab. And we are doing the characterization of new cannabinoid drugs against chronic pain. We are studying it in animal models. And we are a unit associated with the CIC. It is the most famous research center of Spain, and they are giving us some new compounds to do the screening and to do these first steps of the characterization. And my postdoc is about it. We have to keep the sample alive in the organ bags equipment, and we send an electrical input to see how this sample contracts in a visual situation. We can measure until which point they are going to contract. In theory, or theoretically, when we put this new drug, and if this drug is an agonist CB1 or CB2, they will bind to these receptors, and how interacts with muscle cells, the contraction will be lower. So we are going to put an increasing curve of this concentration of the drug, and in every step that we are making it in a non-accumulative way, we will see an inhibition of the contraction. If we calculate the percentage in front of the basal situation, we can make all these graphics. In case that we want to know if they are binding to the CB1 or CB2 receptors and not, for example, to opioids or cholinergics, we are going to use an antagonist for CB1 and then CB2 to see how the contractions is being modified. So we're going to incubate previously with antagonists, and then we are going to put our new drug. If this is a real cannabinoid drug, the contraction will be stable. We're going to compare always with WIN because it's an active comparator that we have. We know how it works. So we are going to expect to have this curve. And when we put the antagonist for CB2 and CB1, we can see that this agonist it binds to both receptors. Last year, I did more than 20 compounds with these experiments. I can show these ones because the others are under political contracts, under lawyer situations, legal situations. So I'm going to show this one. We see that the curve is very similar for the win. So we did the next step to putting the antagonist for CB1 and CB2. If we see at these middle concentrations to avoid specific bindings, we can see that the inhibition is lower in CB1 and CB2 antagonists. So we can say that it has a potential agonist effect to both receptors. In this case, we can see a very similar curve than the other one, but when we put the antagonist, we saw that just the CB2 antagonist has this lower inhibition of the contraction. So this compound, maybe it has a potential agonist effect, but preferably for CB2. In this case, we have another different situation. We are seeing that it binds to the receptors, but in a lower situation, and we thought that maybe it could be an antagonist. So when we put this compound in the presence of the wing, we saw that the curves were different. So we can say, or we can think that it could be it could have a potential antagonist effect. So this technique is very useful to do the first screening, to follow the next steps of the pathway until characterize a new compound. And if one of these compounds has good results, we can try it in an animal model. For example, the other part of my thesis, of my PhD project, is to try one of these compounds in a paclitaxel animal model focused on chronic pain.